Hi everyone, my name is Ajay Reddy, and I'm presenting Chapter 17, The Origin and Early Evolution of Life. So proteins mediate nearly all processes that make up the life of a cell. A few examples of the processes proteins are integral to are the replication and repair of genomes, the control of gene expression, and cell division. Here's a schematic of the process of protein production. Now, all of these steps depicted, replication, transcription, and translation, require proteins. So if proteins are required to produce proteins, how did the first proteins arise? That, the origin of life, is the topic of this chapter. To identify the earliest form of life, we need a definition of life. A minimalist definition of life would be phenomena that are capable of both self-replication and Darwinian evolution. Self-replication means that the system relied only on small molecules and energy for propagation. The image on the top right is a self-replicating machine. Darwinian evolution means that the system was subject to mutation and modification, allowing for the appearance and selection of more complex, variant systems that propagated more efficiently. The bottom right image is a classic example of evolution, Darwin's finches. So what is the simplest life form that exists today? Among contemporary life forms, the simplest free-living organisms are members of the genus Mycoplasma. These bacteria lack a cell wall. One characteristic species has a 580 kilobase genome encoding 500 genes. The significance of Mycoplasma is that their components represent a minimal parts list for protein-based life. Mycoplasma bacteria consists simply of a triple-layered cytoplasmic membrane and the machinery for cytokinesis, chromosome replication and segregation, protein synthesis, and metabolism. These would seem to be the minimal capabilities required for life as we know it. It is possible in the lab to generate bacteria that are similar to Mycoplasma but lack the machinery for cytokinesis. These bacteria are called L forms and divide by a process that is similar to that of the protocells from which life may have arisen. So if those are the minimum requirements for life, when did life appear? Assuming that life arose on Earth and was not brought here in the form of amino acids on comets, we can presume that it could not have arisen before the appearance of liquid water. Earth formed over the course of tens of millions of years. 4.5 billion years ago, a massive collision gave rise to the Moon. The energy of that impact would have melted the mantle of the Earth. Liquid water would then not have existed until it was cool enough for clouds to form. The oceans then began to form no later than 4.4 billion years ago. A stable hydrosphere would have existed about 4.2 billion years ago. And life existed as early as 3.5 billion years ago. The evidence for that is provided by the existence of stromatolites, which are layered structures in the fossil record formed from the trapping of microbial communities in physical sediments. The image is a field of stromatolites in Western Australia. In addition, isotopic evidence dates the appearance of biological carbon and sulfur cycles to this period. That provides a rough estimate of when life may have arisen. A complicating factor, though, in ascertaining the timing of life's origins is that life may have arisen multiple times. Despite this fact, only one life form became what is known as the last universal common ancestor. It is noteworthy and significant that all life on Earth arose from this common ancestor. So how then did this last universal common ancestor arise? Specifically, the building blocks of all life on Earth today are organic molecules. In our protein-based world, these amino acids, sugars, nucleotides, and lipids are produced by enzymes in complex multi-step biosynthetic pathways. This raises the question, where did the building blocks of life come from before there was life? In 1953, Stanley Miller and Harold Urey performed a classic experiment. The question they sought to address was, could conditions on primitive Earth have sustained chemical reactions capable of turning inorganic precursors into organic molecules? You can see the setup in the image on the right. Water vapor and methane, ammonia, and hydrogen gases were combined in the presence of an electrical discharge which simulated lightning. Within a week, a significant portion of the methane was converted into organic molecules, including a racemic mixture of 11 amino acids. Stanley Miller continued this line of research. In subsequent experiments, he included hydrogen sulfide. The result was the production of all 20 amino acids found in proteins today. Only recently have efforts to generate the building blocks of polynucleotides using prebiotic chemistry been successful. Recently, a combination of 4 chemicals that would have been common in prebiotic conditions has led to the production of pyrimidines. You can see the reaction pathway in the image. Microscopic layers of clay are found to adsorb and concentrate nucleotides, allowing them to react with each other to form ribonucleotide chains. This is significant because the most compelling ideas on how life got its start posit the spontaneous appearance in prebiotic Earth of self-replicating RNA molecules. So how did our protein-based world get its start? The discovery of ribozymes provides a potential answer to the mystery of the origins of life. If RNA could serve both as an information carrier and simultaneously as a self-replicase, then life could have started without the need for protein. In this view, mutation and natural selection would have eventually allowed systems to evolve that had the capacity to catalyze peptide bond formation in a manner directed by nucleic acid sequence information. This idea that our contemporary protein-based world arose from an earlier RNA-based world is known as the RNA world hypothesis. So what evidence is there for the RNA world hypothesis? Most of the evidence comes from possible relics of an RNA world. One such possible relic is the self-splicing intron. The image on the top left is a self-splicing intron. The 2 tails in red and blue represent parts of the adjacent exons. Another possible relic is the ribozyme. The image on the top right is a hammerhead ribozyme. It catalyzes reversible cleavage and ligation of an RNA strand. A third possible relic is the riboswitch. A riboswitch is a regulatory segment of a messenger RNA molecule that binds a small molecule, resulting in a change in the production of the proteins encoded by the mRNA. The image on the bottom is a diagram of how a riboswitch functions. You can see that with the binding of a ligand, the ribosomal binding site is hidden from the ribosome. A fourth possible relic is the nucleotide-containing enzyme cofactors that exist. Finally, deoxyribonucleotides are biosynthesized from ribonucleotides. This is consistent with the world in which RNA preceded DNA. Perhaps the most significant piece of evidence for the RNA world hypothesis is the structure and functioning of the ribosome. This image shows the large and small subunits of the ribosome. The rRNA is depicted in orange and yellow, and the protein components are blue. The ribozyme at the heart of the ribosome is entirely responsible for its catalytic activity. The ribozyme acts directly on a carbon center to create a peptide bond. It is significant that what is perhaps the most fundamental reaction of our protein-driven world is catalyzed by an RNA molecule. It suggests that the ribozyme is a molecular fossil from an earlier life form when many or all macromolecular transactions were executed by RNAs. And that's it from me.